Hello, and welcome to the Sleep Science Podcast. I'm Penny Lewis, a neuroscientist specializing in sleep and memory and the presenter of this show. In the podcast, we talk about all things related to sleep, from dreaming and sleepwalking to what sleep does for our brain and body and how we can get more out of our sleep. Today's guest is Professor Michele Bellesi of the University of Camerino, who has possibly done more work on how sleep impacts upon glial cells and brain structure than anyone else in the world. Michele, welcome to the show.
Thank you, Penny, for the invitation.
So it's a real pleasure to have you here today. And as you know, this topic is very far from my expertise, so you're going to have to really hold my hand and walk me through it. I wonder if we could just start with discussion about what glial cells actually are and why they are important.
Glial cells are a group of brain cells. In previous estimations, people thought that glial cells were actually the large majority of brain cells. You know, there is that thought that we were using only 10% of the brain, and that ratio comes from the wrong idea that we had 90% of glial cells relative to 10% of neurons. And actually, new estimations revealed that the number of glial cells and the number of neurons are basically the same, so 50-50. So glial cells, as I said, include several groups of different brain cells. We have astrocytes that are, think about the name astrocytes, is something that has to do with stars. That is because these cells has a star-like shape. And so these cells are important because they can, from one end, they form the blood-brain barrier, and on the other end, they are in very close relationship with synapses. So synapses are these sort of junctions among neurons that allow fast communications among neurons. So this direct relationship with synapses is important for regulating the synaptic function. And then we have oligodendrocytes, another class of glial cells. Those are extremely important because they produce myelin. Myelin is an insulator that is building up around the axons that are the structures that allow the traveling of the signals in neurons. And so the function of myelin is to speed up the signaling along these axons. And recently, scientists found out also that oligodendrocytes are not important only for producing myelin, but the fact that they enwrap the axons somehow is crucial for feeding the metabolic needs of the axon itself. Because, you know, the axons can be very long, they can be distant, very distant from the soma of the cell. So the axon, they need to rely on something else in order to survive. And then we have another class, so it's a very long list, another class that is called microglia. So microglia is the resident immune cells in the brain. So they are basically sort of macrophages that during the early stage of development enters into the central nervous system, invade the nervous system, and there they exert different functions. Of course, they are immune cells, so they get very activated if there is some infection or if there is something that is very strange for the brain. But they are also important, was a recent discovery, for shaping the landscape, especially the landscape of synapses, because they have this remarkable phagocytic activity, meaning that they can eat bacteria, viruses, of course, in case of infection, but they also can eat some pieces of the nervous system, including synapses. So they are extremely important during development when there is an excess of synapses that needs to be trimmed down. And so they are important not only in the pathological scenario, but also in the more physiological scenario. And finally, we have the ependymal cells. So the ependymal cells are another class of glial cells, often neglected. So people usually don't talk about this last class, but they are extremely important because they are the cells that are covering up basically the space that contains the cerebrospinal fluid, the CSF. So it's the liquid that is surrounding our central nervous system. And so basically the function of these cells is to make this CSF moving around because they have cilia, so they have a sort of elongations that they can move in synchrony and make the flux of this CSF moving inside this space. And a special kind of these ependymal cells are extremely important because they are located in the choroid plexus that is a part of the brain in which the CSF is produced. So they are also involved in the production of CSF. So this is in a very nutshell the overview about glial cells.
So that's a really helpful overview of these 4 different kinds of glial cells. So my next question, I think, is way too broad, but so what happens to these glial cells in sleep?
Yeah, that's a very good and very interesting question. It's very difficult to answer because sleep and wake and eventually sleep loss can affect the function of these cells in different ways. So basically what we did to understand the effect on sleep and wake on these glial cells was to use some sort of special methodological approaches that rely on studying the transcriptome of the cells. So the transcriptome is basically every cell has its own DNA that is used to store the information about the cell. So this DNA is also used to create proteins. That is the other level of information that we have in the cell. So basically the transcriptome is a way to understand which proteins are going to be produced given that original DNA code. Okay, so if you want to understand which functions are shaped by some external factors, by some drugs, or in this case about sleep and wake, studying the transcriptome is one of the best way because it can give you the broad view of what's happening inside the cell because of sleep or because of wake. So basically what we did is we studied the transcriptome in this specific cell types. We started with the oligodendrocytes. We found out that sleep somehow is important for stimulating the production of proteins that are implicated in the myelin production. These are the most functions of oligodendrocytes that are promoted by sleep. Vice versa, it means that if you are reducing sleep or if you are in a wake state, of course you have the opposite. You have a reduction of myelin production, a reduction of lipid production that are used for creating this insulating mechanism that is actually the myelin itself. So, for example, in this specific case of the oligodendrocytes, we found out that the sleep is actually important for myelination, for producing new myelin. This then has been confirmed by other studies in animals, but also in humans. In animals, for example, we were able to measure the extent of this myelination by physically measure using electron microscopy the extent of the myelin surrounding the axons, and we found exactly that if you deprive the animals from sleep, you notice a decrease of this myelination, meaning that sleep is important for this cellular function. This study has been confirmed also in humans. In humans, of course, you cannot do electron microscopy in living humans. You need to rely on indirect methodology. In this case, for example, if you do structural MRI, there are, I think, so far a number of different studies coming from different laboratories, which almost all show that indeed some markers of myelination are reduced in people sleeping less or in people with bad sleep quality. Okay, again suggesting that sleep probably is important for boosting myelination in the brain. If we look in astrocytes are implicated in different brain functions. So the first thing that we discovered is that in general, these cells seems to be very active during wake, meaning that they are producing a lot of proteins relative to sleep. But that doesn't mean that their sleep is not important for these cells. So in wake, it seems that astrocytes are particularly important in sustaining synaptic activity. As I said before, these astrocytes, they can have very long processes, they can reach the synapses, and there they can do several things. They can, for example, help remove the excess of a neurotransmitter, that is the chemical signal released by neurons in order to evoke the signal in the postsynaptic cell. So they are implicated in removal the excess. So in a way they can shape synaptic transmission by doing that. They can they can balance the ion environment. You know that all these electrical signals created by neurons rely on an imbalance, on a transient imbalance of ions. So ions concentrations must be very well regulated. So astrocytes, thanks to their ion channels, they can regulate this very precisely, especially at the synaptic level. And also they can sustain the metabolic activity of the synapses by providing, for example, some metabolic substrates that synapses can use to sustain their activity. Keep in mind that synaptic activity is one of the largest metabolic costs for the brain. So it's very important that synapses are in a place in which they can be fed very well. Finally, it's been recently discovered that astrocytes, again, like microglia, for example, they can shape the structure of the synapses by, for example, eating a piece of synapses that are probably damaged. And this is something that we have studied also in our lab in a very detailed manner. For example, lack of sleep, especially if the lack of sleep is prolonged, astrocytes starts to eat portion of synapses. The reason why this happens is still unclear, but probably is related to the fact that synapses, like every other structures in the brain, needs to be somehow maintained, okay? Because membranes forming these synapses can get oxidated, portion of the cells can get ruined by the fact that are used quite a lot. And so there must be some sort of mechanism to replace what is wrong, what is getting oxidated, getting damaged. And so one of the possibilities that astrocytes are also implicated in this phenomenon. In fact, in mice in which lack of sleep was particularly pronounced, we found a very pronounced effect of this phagocytosis of astrocytes, meaning that this sort of turnover of cellular membranes of the synapses was much higher. And that probably one of the function of sleep itself is to maintain a correct environment at the synaptic level. Finally, if we look at the microglia, again, microglia probably behaves differently in wake relative to sleep. Although the situation here for the microglia is a little bit more complicated because the studies are showing different things, meaning that there is not a uniform view about what's happening to microglia during sleep and wake. For example, there are studies in which have shown that also microglia can send these tiny processes nearby the synapses, and some studies have shown that this happens mostly during wake, whereas other studies have shown that this happens mostly during sleep. So there might be a little bit more controversial situation for microglia. Finally, the last class, the neglected one, the ependymal cells, has been very little investigated in relation to sleep and wake. We know very little probably about their circadian modulation, meaning that, for example, the CSF production is occurring more during the night in rodents and in humans. So somehow there is some sort of circadian regulation. But you know that in humans, sleep is occurring mostly during the night period, whereas is the opposite for the rodents. So this somehow disentangle the role of sleep in the CSF production. But of course, there are much less studies for these ependymal cells, and we actually don't know if their function is different across sleep and wake or in different behavioral states.
Yeah, and it's amazing that you are able to synthesize that all together for us. Let me see if I kind of got it. Working backwards, so the ependymal cells, you say that they produce more fluid at night, and that's actually the wake time. So they are producing less fluid probably during sleep.
No, not exactly. I mean, this is just one study, and it shows that basically in both humans and rodents, there is more CSF production during the dark period. Okay. The problem is that in humans, the dark period corresponds more or less to sleep, whereas in rodents, the dark period corresponds to a moment in which animals are spontaneously awake. It means that yes, probably there is a circadian modulation of CSF production, but I don't know, or we don't know, how much this modulation is actually also related to sleep and wake.
I see. So it's complicated, basically. There's that kind of unclearness there. And those are the less studied ones anyway. So the microglia, so they send processes to synapses, and this seems to happen less in sleep. So they are modulating the synapses possibly less, or they're influencing the synapses less in sleep.
I mean, this is again, it's another gray area because there are studies that show this is indeed the case. In sleep, you have less processes around the synapses, less microglial processes. And this is again what also we showed, not in relation to sleep and wake, but to sleep loss, which is a little bit different than spontaneous wake, at least in the animals. So, for example, in our study, we found that if you deprive the animals from sleep, you tend to see a retraction of this process. Because microglia need to move around, tend to become, it's called the amoeboid state. So it becomes with less processes so that they can infiltrate better into the brain parenchyma. So that's why there is this belief that the structure of microglia somehow can tell you about the function of microglia. So microglia cells that are with a lot of processes, with a very complex ramification of the processes, are probably microglia cells that are there, they are still, and they are surveilling the parenchyma. Okay. Instead, if you want to have a microglia cells moving around in the parenchyma, then you need to retract these processes and become more simple in terms of complexity of the processes.
Why do they need to move around when there's sleep deprivation?
Because they follow some cytokines, some factors that are released by cells that are suffering, cells that are dying, or by bacteria, by viruses. So you need to have in the brain a system in which the immune cells can move to a place in which there is something going on that is wrong. In more physiological terms, when there is, for example, damaged cells, I told you before about the damaged synapses due to the high-intense activity that mostly is occurring during wake. So microglia can get activated by these signals that are called hit-me signals. So they are signals released by the cells, neurons or other cells, that are triggering the microglial motility because are signals of alarming. And so these hit-me signals are calling the phagocytic cells that are microglia in most of the cases, but we have seen also that astrocytes can do that. They come around and they start eating the damaged part of the cell. Or if the cell is completely dead because it underwent to apoptosis, it can happen in the brain. So, of course, you have to remove that cell. So microglia moves to that point, eats the dead cell, and then clear basically the space from something that is no longer used. So microglia needs to move inside the brain. So that's why it can lose this complexity in the processes and move around. Although there is not a uniform agreement on this reduction of increasing of complexity in across sleep and wake.
Okay, so what you mean is that in the sleep-deprived condition, you see that they've retracted their processes so that they can move, and therefore you know that this kind of moving around and eating the other cells is happening, but you don't see that they've retracted them so much if you just sacrifice the animals during a sleep period. So it's when they are sleep-deprived.
Exactly. Plus, there are differences, for example, between the model that you are using. For example, there are differences between rats and mice. Okay, so microglia in rats seems to be less sensitive in terms of structural complexity between sleep and wake. Mouse microglia is it's very sensitive to sleep loss. For example, if you do even 4 hours, 5 hours of sleep deprivation, you can immediately see that the structure of the microglia changes. also there might be regional differences. Okay, for example, in some areas of the brain, there might be more. This phenomenon might be more evident. In other brain regions, it might be less evident. So it's still a matter of debate whether these processes are really extending and retracting according to sleep and wake, and of course their relationship with the synapses. It's not easy to study. It's also not easy to draw final conclusions.
Are there ways that we can actually visualize them moving, or is it always sacrificing the animals at different time points and sectioning and staining and can we see it happening in vivo, or is it always?
So yes, it's possible, but the movement doesn't occur in seconds. I mean, it's something more slowly. So you need to observe the in vivo tissue for a longer period. And this is not always super achievable in animal research, because you need to keep the animal alive under the microscope. The animals should be in a behavioral state or in anesthesia. So it's a tricky experiment. But for example, people have observed using this, they can track the movement of mitochondria inside the cells. Okay, so you can really see how the mitochondria are moving. So it's doable to see things that are moving in the living brain, but those are pretty tricky experiments. It's not that easy.
Yeah, to me, it sounds like a fairy tale, like the idea of these cells moving around and eating other cells. And yeah, it's amazing because it's not something I know much about. Okay, one more question on these microglia. Why do you think they are triggered to move more in sleep deprivation? So I understand it's when there are cells that have damage and they need to be cleaned up and eaten. Why should that be happening more in sleep deprivation?
Because in sleep deprivation, usually you might have more the chances to get damaged membranes, damaged proteins. So the cells are experiencing more damage because are more heavily used in wake, and especially in extended wake relative to sleep. We know, for example, the metabolism is higher in wake relative to sleep. In general, the synaptic activity is very high in wake. And when you have a condition of extended wake in which you are really pushing the system a lot, and then you increase quite a lot the oxidative damage, for example, of the membranes, of the lipids, and all these parts must be somehow adjusted or replaced. So that's why these supporting cells starts to engage microglia, but also astrocytes. Because, another thing that is going around, especially with the extended wake, with the sleep loss, it's the increase of neuroinflammation. This neuroinflammation is another thing that are now very well studied because it's relatively important. So it seems that the neuroinflammation, it becomes higher with sleep loss in general. And so all these triggers are promoting factors for microglia to be activated.
So it's a state of physiological stress. There's a lot of use, and also the regular cleaning up and probably support that might have happened to those cells when, the animal should have gone to sleep hasn't happened. And this all leads to the kind of damage that you're talking about, right?
Yes.
Okay, so that's the microglia. Now for the astrocytes, you said, again, this is one where during sleep deprivation, they eat the synapses. Is it just the synapses that they tend to clean up in that way?
The astrocytes, in general, all the phagocytic cells, including in this case also microglia, can eat also other structures, not only synapses. For example, they can eat mitochondria along the axons. But the synapses are extremely important because it's the place where the signal is conveyed and transmitted from one cell to another. So are very critical points. And so synapses must work in a very efficient manner. And so that means that need to be in a very good shape always. That's why there are all these players moving around, making sure that the synapses are fine. And so the astrocytes, since are there with their tiny processes, they are probably the best suited cells to surveil and do this in case of damage. For example, what we observed, it's not only the fact that the astrocytes start doing this sort of phagocytic activity of synapses with wake periods, spontaneous wake, short sleep deprivation, or long sleep deprivation. They also to move and retract their processes. And this has been studied with three-dimensional electron microscopy, so a very powerful technique, in which we saw that during a wake, these processes are closer to the synapses. So synapses tend to be more surrounded by these astrocytic processes during wake relative to sleep. So in sleep there is a sort of a retraction of these processes. The reason why this happens, is not clear. It might be that the processes are getting there because there is more need for them to be there, meaning that, as we said, the synaptic activity is very high in wake periods, and then there is more need for the astrocytes to be around because they need to recycle more neurotransmitter, they need to rebalance better the ion environment, so they need to do this phagocytic activity, so there is more need for the astrocytes to be around. Whereas in sleep, probably there is less need. Another explanation could be that, in general, in sleep, you can observe this retraction of astrocytic processes. This retraction may favor the interstitial clearing that is occurring during sleep. You know that there is this idea that during sleep there is an increase of activity of this so-called lymphatic system, that is the system somehow that is believed to clear the extracellular space from the waste products that cells, of course, produce during their activity. And so it has been shown that this lymphatic cleansing function of the lymphatic system is more pronounced during sleep. And one of the reasons that during sleep there is this retraction of the astrocytic processes favoring an enlargement of the extracellular environment that is needed to really wash out what's inside. So all these waste products. This is another explanation for why you have this retraction of the astrocytic processes. Another possibility is, but it's never been tested formally, so it's at the moment it's just a speculation, but since the astrocytic processes around the synapses are important for controlling the concentration of neurotransmitters, so if you have less astrocytic processes, the neurotransmitters can spill out outside the synapses, entering into the extracellular parasynaptic space and influencing the other synapses as well. In a way, this might favor, for example, the level of synchrony of activity that you have synapses that are close to each other. And so somehow, if you think that there is a lot of synchronization of electrical activity during a REM sleep, for example, this retraction of astrocytes and the spillover of the neurotransmitters, it might be involved in helping out this sort of synchronized activity among a group of cells, because there is this increase of spillover. So there might be different reasons why you have these retractions. But again, I would like to emphasize that this retraction has been observed using this three-dimensional electron microscopy, and comes along also with other changes within the processes of the astrocytes. For example, this is another thing that is happening in these processes. You know that these processes are containing sort of storage of glucose in the form of glycogen. So the glycogen in the brain is basically present only in the astrocytes. It's a sort of storage of energy. It's a sugar that is condensed together, forming these granules of glycogens. And so basically, these glycogen granules change their distribution, their size, their density according to sleep and wake. So it seems, for example, that during wake periods, there is an increased turnover of this glycogen, because probably synapses are consuming a lot of energy, so there is the need for the astrocytes to free up somehow the glucose, perhaps shuttling this sort of metabolites in the form of lactate to the neural structure to sustain the increased firing during wake. The presence of these astrocytic processes are also for supporting from a metabolic point of view the synapses, not only for recycling the neurotransmitter, balancing the ions, and doing this weird thing of phagocytosis.
Okay, let me see if I got some of that. So what you're saying is that they tend to retract their processes during sleep. They're not just phagocytosing the synapses, although they can do that in sleep deprivation when it's needed because there was damage. And they're retracting. It may allow more flow of the lymphatic system. So it may allow creating space for the fluids to move around. And it may also mean that they are allowing some of their neurotransmitters to leak out, and that can kind of enhance the local synchronization at the synapses around where they are. And those are the two main functions that you said, I think.
Yes.
Okay. Because they are so small and their processes are so small, surely they are not the main ones that, okay, they pull in their processes, but is that really the thing that makes the space for the lymphatic system to move around? Because I guess other cells would have to shrink as well in order for that to happen, not just the astrocytes, right?
Yeah, but the astrocytes have the capability of shrink very much. And this has been demonstrated in pretty well. If you look them under the microscope, they really look like sponges. And so they can quite a lot retract their processes and then generate, of course, space for the extracellular environment, favoring the lymphatic system.
And when they do that, does the cytoplasm or the fluid in their cytoplasm, does it go out? So they're actually shrinking in terms of the amount of fluid they contain?
Yes, by regulating the amount of ion, thanks to the presence of aquaporin that are channels for water, they can change quite a lot the amount of cytoplasm and water that they have inside. So that's why they are the candidate cells for the lymphatic system because of that, and also because they are forming the blood-brain barrier on the vascular side, and they are connected to these perivascular structures in which supposedly fluid, the CSF, is moving around and helping out the clearance of wasting products.
And do we know if the shrinking is more, do we know what stage of sleep? Do we know, is it non-REM or REM sleep?
I don't know. Theoretically, it should be more in the non-REM stage, meaning that probably it's where you have most of this shrinkage. But I don't have knowledge of the fact that this has been studied in so precise manner, isolating non-REM versus REM. Because the way you do the differential function between REM and non-REM is tricky in the animals because you need to manipulate non-REM without not manipulating REM, and they usually go together. When you do sleep deprivation, you have, of course, a reduction of non-REM and REM as well. It's very difficult to do REM deprivation alone without affecting non-REM, so it's not that easy to study this phenomenon.
That was my impression, is that people don't really distinguish this because it's hard. It's hard to study. So, yeah. Okay, let's go back to the lymphatic system. Maybe this is a good time to tell that story because we've touched on it several times. And can you maybe introduce the story of the lymphatic system and the kind of mystery or debate around that question? So
basically, the lymphatic system is something that it's a sort of an hypothesis generated by Mike and Nedergaard group, sustaining that in the brain, there is lack of proper lymphatic system as in other organs. So there must be some way to clean and remove the waste products. So they come up with this idea of the lymphatic system, and they studied in different ways, in vivo in the animals. They have indirectly assessed in the humans thanks to functional MRI. And so what they found is that specifically during sleep, there is a sort of an enlargement of the extracellular space, and there is an increased clearance of this space thanks to the movement of the extracellular liquid, that is the liquid surrounding all the cells, from periarterial space, that is the site where the blood vessels enter into the parenchyma. These arteries are surrounded by a sort of periarterial space that contains a little bit of CSF. So that CSF can move through, thanks to the astrocytes, into the extracellular space and then be recollected again into perivascular space of the veins. So this movement of fluid from the arteries to the veins through the extracellular space has the function to clear up the toxins, the waste products of the cells that are present in the parenchyma, and seems to be more active during sleep because of this retraction of the astrocytes, because astrocytes are working to promoting this sort of fluid movement during sleep. Although recent evidence by other laboratories have shown with different techniques actually that this clearance of the brain parenchyma is actually not higher during sleep but during wake. And so at the moment there is this debate on whether this lymphatic system is clearing up the extracellular space more during sleep or more during wake. And the answer, of course, is probably in the middle, or we still don't know. We need probably more evidence to see which of the two sides correct. There are, of course, evidence not coming only from this Mike and Nedergaard laboratory. There are evidence from other laboratories. There are evidence even in humans showing that the CSF seems to pulse, especially during slow wave sleep. So somehow the slow waves generated by the brain are involved in the movement and the pulsation of this extracellular liquid throughout the parenchyma. Recently, the Mike and Nedergaard group proved that this movement of the lymphatic system is actually promoted by the activity of the locus coeruleus, that is a brain structure in the brainstem producing noradrenaline in such a way that the production of this noradrenaline is not produced regularly but somehow is oscillating following some infraslow rhythm of the brains, and this infralow oscillation seems to be promoting the release of noradrenaline in the brain and also triggering this contraction of the arteries, promoting then the production and the clearance of the lymphatic system. So it's a very interesting story that is growing day after day, and it's very nice to see how we'll end up. It's very important because this lymphatic system is not only be associated with sleep, but is actually being studied at the beginning in relation to the accumulation of beta amyloid, that is one of the leading cellular hallmarks of Alzheimer's disease. So they show that, for example, during sleep you have a clearance of beta amyloid from the extracellular space, and perhaps this is important for the Alzheimer's field in general, for the neurodegeneration. And we have actually a recent publication in our lab in which we enhanced sleep in mice by rocking them with vestibular stimulation and in a mouse model of Alzheimer's disease, and indeed we found by boosting sleep a reduction of beta amyloid in the extracellular space. Mm-hmm. So we don't know if this reduction of beta amyloid is related to a more active lymphatic system or just a less activity or less production of a beta amyloid from neurons. We didn't test this directly, but again this goes into the direction linking the importance of sleep with several diseases, especially neurodegeneration.
Mm-hmm. I mean, I think one of the reasons that, that this is a topic of emotional debate is the link to degeneration, possible Alzheimer's disease, and also the fact that this work on the lymphatic system has led to a lot of work in humans trying to manipulate sleep largely by boosting slow oscillations with the idea that that could slow down, some of this deterioration and into Alzheimer's disease or just even kind of brain aging. I think this is why people get worked up on this topic, right? So you mentioned a couple of times the pulsing with the slow oscillations and also with the locus coeruleus and the noradrenaline. So it does seem that this has been linked to slow wave sleep, or at least non-REM sleep, rather than just sleep in general, right?
Yes, specifically the portion of sleep in which you can read very well this infraslow oscillation, which probably in animals is throughout the entire non-REM sleep. Perhaps in humans is more related to the moment in which you have more spindles, that is, for example, N2. So this is another interesting point, is to understand whether the entire non-REM sleep is characterized by this infraslow oscillation or mostly lighter stages of sleep, at least in humans. What what is very interesting is this relationship between the locus coeruleus activity that is no more stable during non-REM, because at the beginning people thought that yes, noradrenaline is higher in wake relative to sleep, and this is of course true. If you look, for example, at the old studies in which they measure with microdialysis the levels of noradrenaline in the cortex, we did it also with Chiara Cirelli and Giulio Tononi in Madison. So indeed there is a difference in the terms of on the levels of noradrenaline between sleep and wake. But what is been found recently is that the locus coeruleus activity itself is not completely silent. I mean, it's pretty silent during REM sleep, but during non-REM sleep is actually oscillating, creating this moment in which the sleep is more similar to wake and moments in which sleep is deeper. So with this different fragility of sleep oscillating in the non-REM sleep. And this is super fascinating, and is also even more fascinating the fact that this has been linked to the lymphatic system, to the fact that this oscillating driver of the noradrenaline can really play a role in the movement of liquids, movement of fluids into the brain. I mean, this is, I think it's very exciting.
And would this be because the astrocytes are pulsing as well? I mean, how does the noradrenaline mediate the movement of...
The overall idea is, I mean, at least for this latest publication, is that the oscillating activity of locus coeruleus somehow is stimulating the arteries, so somehow the muscular activity around the arteries, so that they can really pump the blood in a way and produce this movement inside the brain parenchyma. But we know from other studies that also astrocytes are very sensitive to noradrenaline, so it could be possible that even astrocytes are sensing this infraslow oscillation of noradrenergic activity in a way that helps the lymphatic clearance of the extracellular environment. Although I don't think that there is still direct evidence of that, but that would be a nice confirmation.
And this pulsing of the locus coeruleus, you said it's infraslow, so that's every several seconds, right? Like 4 or 5 seconds?
No, it's longer, the period. I think it's around 15 seconds.
So that's very different than the period of the slow oscillation, because there was also a discussion about whether the slow oscillations could cause some kind of a pulsing. I don't know how that would work.
I don't know. I mean, I don't think that this has been clarified. But for example, in the latest paper of Mike and Nedergaard, the one I was referring, linking the locus coeruleus activity and the lymphatic clearance, for example, they didn't find such a huge correlation with delta. So it seems more that this infraslow oscillation of the locus is what is really driving the pulsing of the lymphatic system more than the slow waves, at least in this animal model. But, you know, maybe that it doesn't exclude the fact that even the slow waves itself can play a role. After all, you are packing the activity of neurons in 1 second at a time, you know, with the slow waves. And so somehow you are creating some sort of oscillating environment in which you have neuronal firing and neuronal silence. And this somehow, I'm sure that has an effect on the way the fluids are moving in the extracellular space.
So, I mean, it's ongoing work. It's super fascinating. Watch this space, basically, is the message on this. Let's move on. So the last cell type that we didn't come back to was the oligodendrocytes. And what you said about them with respect to sleep, I think, was the myelination actually decreases with sleep deprivation. Can we talk a bit more about this? Why would the myelination decrease when they are sleep deprived?
Okay, so here we have direct evidence measured again with this electron microscopy. We observed that if we do sleep deprivation, there is indeed a decrease of the thickness of the myelin, that is again this sort of fatty substance created by these oligodendrocytes to insulate the axon from the extracellular environment, so that the speed of the action potential traveling in the axon can move faster. It's something that we acquired later in the evolution and guaranteed us to have fast spiking signaling in the central nervous system without increasing too much the size of the axons. So myelin is some clever device that nature invented to allow the fast propagation of the signals and allow our cognition, basically. That's why these cells are super important. And the fact that the extent of myelination can change between sleep and wake is also super fascinating, because myelination can regulate the speed which the axon potential is traveling. And so in a way, the oligodendrocytes has the power to decide the synchrony, the timing of arrival of the single spikes into the nervous system. And so that is great power, because it can control if the signals are arriving in synchrony or if arriving with some delay. So by controlling myelination. And so what is been found in the latest years is that myelination is not stable as we thought it was. It's very dynamic. It can be regulated according to the need of the neurons. For example, if you are learning something, then the circuits that are underlying that new skill gets myelinated quite a lot, because the organization of the signal, how it travels along these nerves, must be very well regulated. So the oligodendrocytes can display these plastic functions that were completely neglected a few years ago, that are coming up now. So in fact, people discuss about the myelin plasticity. We are used to hear the term synaptic plasticity, but now we have also myelin plasticity, because by modulating the strength of myelination, you can really impact how the signal is traveling, and therefore the synchrony, the performance of that local network of neurons. So what we found is that, especially during sleep deprivation, there is a reduction of this myelin. And the reason why it happens, we believe, is that the oligodendrocytes has to prioritize some functions. So basically, if we want to simplify the things, we can say that the oligodendrocyte has two main functions. The first function is to create this insulation through myelin. The other function is to feed the axon that is insulating. Somehow, during sleep deprivation, you have an increased firing. So this intense firing requires a lot of energy for the axons. And so the oligodendrocytes shift towards feeding the axon, then creating more myelin.
Is that in sleep deprivation?
Yeah. So this shift occurs in sleep deprivation, in which you have a greater need for the neuronal structure, the axons, to do the fast spiking, to generate action potential. So the axons need a lot of energy. And so this energy is coming down actually by the oligodendrocytes. So the oligodendrocyte has to make a choice. Do I have to produce more myelin, or do I have to sustain and support the axonal metabolism? So in our opinion, the shift is occurring right there. So the oligodendrocytes during sleep deprivation prioritize the axonal support relative to the myelination. So that's why we have a decrease of myelination. There is also the idea that myelin, since it's very rich of lipid, it's a sort of an energy storage for the brain, because you can remove the lipids, and then these lipids can enter in particular metabolic pathways that can produce energy at the end. So this is also probably true. In the case of starvation, in case of more energy expenditure, the oligodendrocytes decides to reduce the extent of myelination in favoring the metabolic support for the axon.
They're favoring keeping the system healthy over having it optimized and fast. And so this is in sleep deprivation or in other states of stress. But what about during sleep? Do we know how their behavior differs in sleep?
What we believe is that sleep is a good moment instead, in which you have probably less energy requirements to build up cellular structures. And myelin in general requires a lot of building up, because you have to pack together proteins with a lot of lipids, with a lot of cholesterol, and all this stuff. If you look, for example, at the transcriptome, it's pretty well evident that during sleep you have an upregulation of all these genes that are implicated in forming new lipids, new proteins. So because it's a good moment to build up stuff, and that's why we believe that sleep is important for myelination, because it's a good moment in which the axon probably has less energy requirements, it's less strictly dependent on the oligodendroglia that is free to engaging in myelin production.
So it's getting all the stored supplies ready for the day when it will be active and it needs to use those things. So building up supplies, basically.
Exactly.
That makes a lot of sense. Now, I'm aware that we are running low on time, and it's frustrating because there's so many things I want to ask you about. But I feel like we covered, for me, it was a huge amount of information, but I also feel like we're just scratching the surface, and you are opening up this window on this whole world of a brain where cells move around and interact and care for each other and clean each other up, and all of these kind of dynamics that I'm not very aware of. So it's completely fascinating, and I could talk to you all day, but I realize we don't have a lot more time. Is there any other area that you feel like we should have talked about, an area of your work that you would really like to mention that's exciting?
Well, the other line of research of the lab is, of course, this entire story of sleep enhancement that we are always fascinating about, because it's a completely new way to study sleep. People usually use sleep deprivation to discover the function of sleep. So you deprive an animal, a human, of this biological function, and then you see what's going on, what's wrong. And so you infer that the function of sleep then must be related to that. But sleep deprivation comes with a lot of caveats. You have stress, because it's impossible to do sleep deprivation without stressing the animal or stressing the humans. So at the very end, you never really disentangle the stress from the sleep deprivation. You don't really know if what is really the function that you are discovering is really truly related to sleep or the stress of what you're doing to the person or to the animal. The sleep enhancement in a different way. So sleep enhancement can be done either pharmacologically, but it's something that again comes with sort of it's not really a clear experiment, the drug itself, because there are effects directly related to the drugs that might be not related to the fact that you are enhancing sleep. So I like very much the idea of using sensory stimulation, which is in a way what you are doing as well, by using acoustic stimulation, or for example we have seen that in animals, in mice especially, the rocking is a very strong sensory based sleep enhancement method in which you gently rocking the animals. You see that this has an important effect on especially on the duration of non-REM sleep. You get more non-REM sleep. The animals tend to fall asleep more quickly. The periods of sleep are longer, so there is less fragmentation. So I think this is a good model to study again the function of sleep by trying to boost what sleep does in the brain. So it's a different approach, a different methodology, but assessing the effect of sleep without the problems that comes with the sleep deprivation.
I mean, you know that I agree, because this is also the approach that we take. But I think it's great that you're doing this in rodents.
Yes, in fact, because there are not many laboratories that are studying these sleep enhancement methodologies applied to rodents. You know that in humans, acoustic stimulation has been used quite a lot, even with controversial results related to the fact that, you're pushing probably a little bit the arousal system. So you are always on this silver lining in which you are pushing the sleep slow waves, but on the other end, you're using the system that nature gave us to be awake. So somehow can be very tricky to use acoustic stimulation. And this is true also for the animals. But again, it's a different approach to analyze the function of sleep, a different way of what usually people do, which is the use of sleep deprivation.
And have you been able to extend our understanding of any of these different cell types that we discussed using this sleep enhancement?
This is actually something that we are doing at the moment in the lab. I cannot spoil the preliminary results. But yes, we can really boost some cell functions by enhancing sleep. And of course, you boost the functions that are sleep dependent in each different types of different brain cells. So for example, we know that sleep is important in neurons for synaptic plasticity. So if you do that, then you promote synaptic plasticity, you promote learning. Whereas if you look at oligodendrocytes, then you see an improved myelination. And then if you look at the astrocytes, you see something completely different. Again, it's a nice way to disentangle the role of sleep in different brain cell types.
Yeah, very nice way. Well, it sounds like we need to watch this space, because you said you're not going to spoil it here, but it sounds like you have some very exciting results that we'll see coming out on this soon.
Yes.
Yes. Okay. Well, Thank you so much for explaining all of these things and for telling me about your wonderful research. Yeah, it's really amazing to hear about this.
Thank you. Thank you.
You've been listening to the Sleep Science Podcast with me, Penny Lewis, and my special guest, Michele Bellesi from the University of Camerino. From me and all the researchers at NAPS Lab Cardiff, thanks for listening and sleep well.